नहमदु व नुसल्ली अला रसूलिहिल करीम अम्मा बाद फ़अऊज़ु बिल्लाहि मिनश शैतानिर रजीम बिस्मिल्लाहिर रहमानिर रहीम. Assalamu alaikum to my respectable teacher and all my fellows. Aisha Rashid warmly welcome you all in her presentation. Dear students, hope you are all doing great. The video on this topic contains instructive and educational content that are both shareable and easy to retain. So let's start. My presentation topic is receptors physiology. First we discuss about receptors. What are receptors? If we discussed in physiology, a receptor is any structure which on receiving environmental stimuli produces an informative nerve impulse. Receptors in biochemistry. In biochemistry, a protein molecule that receives in response to a neurotransmitter or other substance. Now we discuss about the classification of receptors. There are four different types of receptors. The first one is liquid-gated ion channel, which is also known as ionotrophic receptor. The second one is G-protein coupled receptors, which is also known as metabotrophic receptor. Third one is kinase linked receptor, and the fourth one is nuclear receptors. Here is the mechanism of these different receptors. At the bottom of this diagram, a table shows different time scales and example. This is all about the classification of receptors. These are my subtopics. Now we discuss about the transduction. If we take it wordly meaning, transduction means conversion from one form to another. Sensory transduction is a conversion from a sensory system or sensory stimulus from one form to another. Now we discuss the diagram. In this diagram, the gray line shows plasma membrane and the purple one is receptor and the yellow one is environmental stimulus. Environmental stimulus may be a hormone, ion, inorganic substance or protein, enzyme, anything. When the stimulus apply or act on a receptor, this process is known as reception. The second process is signal transduction, which is act on in a cytoplasm. When reaction producing second messengers, this process is known as signal transduction. According to the receptor or stimulus, some response are produced. This is the last process. This is all about the transduction. Now we discuss about the sensory transduction. In sensory transduction, receptor transform an external signal into a membrane potential. There are two different types of receptor cells, a nerve cell and a specialized epithelium cells. In the diagram, we discuss the structure of stimulus. How calcium-gated ion channels allow calcium ion to release transmitter. These transmitters attach on an efferent neuron, and this diagram also shows the direction of action potential. Now we discuss about the sensory coding. Sensory coding is a type of information processing that occurs in the nervous system and can be thought of four different phenomena. The first one is modality, the second one is location, and the third one is intensity, and the fourth one is duration. If we discuss modality, modality is a type of energy transmitted by the stimulus. In the diagram, it shows how the energy transmitted by the stimulus. Second one is location. Location is the site of the body where the stimulus originated. There are four different types of fingers that shows how we found out where the stimulus originated. Third one is intensity. Intensity is the response, amplitude or frequency of action potential generation. And the fourth one is duration. Duration is the time from start to the end of a response in the receptor. This is sensory coding. Now we discuss about the range fractionations. Range fractionations is a term used in biology used to denote range-finding threshold for different stimuli intensities. Sense organs are usually composed of many sensory receptors measuring this same property. Here at x-axis we show log stimulus intensity, and the y-axis we show frequency of APS impulses. There are four different types of curves. The first curve shows the range of one receptor and all the curves shows the range of a system. This graph helps to find out the value of range fractionations. Now we discuss about the sensory adaptation. Sensory adaptation is a gradual decrease over time in the responsiveness of the sensory system to a constant stimulus. It is usually experienced as a change in the stimulus. For example, if a hand is rested on a table, the table surface is immediately felt against the skin. There are six different graphs that shows how gradual decrease occur in the adaptation curves for sensory receptors. This is all about the sensory adaptation. Now we discuss about the mechanoreceptor. Mechanoreception ability of an animal to detect and respond to certain kinds of stimuli. For example, notably touch, sound, and changes in the pressure and the posture in its environment. If we discuss the diagram, a reflex hammer shows a stimulus where sensory and motor neurons carry the message to the spinal cord and spinal cord responds according to the stimulus. Now we discuss about the hair cell mechanism, particularly in the acoustico-lateralus system of vertebrates. This is a specialized system. In this system, the inner ear region and the lateral organ located in the skin forming a sensory system that conveys environmental information to the brain of the fish. The lateral organ respond to the changes in water pressure and displacement, and the inner ear respond to the sound and the gravity. In this diagram, the outer hair cell and the inner hair cells shows how acoustico-lateral system work. Now we discuss about the cutaneous receptor. Cutaneous word is related to the skin. The cutaneous receptors are the type of receptors found in the dermis and epidermis. They are part of the somatosensory system. Cutaneous receptors include cutaneous mechanoreceptors, that is nociceptors, and the thermoreceptors. Nociceptors are specialized for the pain, and the thermoreceptors are specialized for the temperature. In this diagram, some dermis cells, epidermis, root hairs, follicles, nerve endings, Pacinius cells, free nerve endings, Merkel's disc cells, root hair plexus, different cells shows the cutaneous receptor. Now we discuss about the olfactory reception. The word olfactory relates the smell. Olfactory receptor is also known as smell receptor protein capable of binding odor molecule that plays a central role in the sense of smell. The cilia are conveyed by the mucus of the nasal cavity, facilitating the detection of the response to odor molecule by olfactory receptor. If we found out a little review on this diagram, the olfactory bulb is zoomed in where glomerulus and nerve endings show how they relate each other. Olfactory epithelium. In this diagram, we discuss small changes in a single olfactory receptor gene can affect how we feel odor. When a person smells a flower and olfactory receptors nerve receives some olfaction, these odorant molecules and the nerve ending of these neurons binds and respond to it. Now we discuss about photoreception. Photoreception refers to the mechanism of light detection that leads to the vision and depend on specialized light sensitive cells that are called photoreceptors, which are located in the eyes. Some light sensitive cells are rods and cones. This is the structure of an eye. Now we discuss about the ultrastructure of photoreceptors. There are two types of photoreceptors, rods and cones. Here it is the structure of rods, how rod look like. And here it is the structure of cone. A cone-like structure is known as cone cells. In this cone cell, when we found out the structure of rods and cones, we discuss lamella, outer segment, connecting cilium, inner segment, mitochondria, nucleus, and myoid synapse. Now we discuss the outer segment of rods. In outer segments of rods, we discuss some important points: lamellar particles, free-floating discs, ciliary fibrils, limiting membrane, folding and outer cell membrane, connecting cilium. This is the structure of rods and cones. Now we discuss about the physiology of color vision. Here it is a flow chart form in which we discuss photochemistry of color vision. When we capture a photon, the irreversible photochemical isomerization of chromophores occur. Series of photon conformational changes occur. There are two types of reaction: active pigment or catalyst. When in catalyst, a transducing protein molecule is in active form, where cGMP phosphodiesterase decrease by level to level. At the end, hyperpolarization of the photoreceptors. This is the physiology of color vision. My last topic is physiological mechanism in electroreception. Electroreception relies upon the tubulous electroreceptors, which are sensitive to high frequency stimuli, which is 20 to 20,000 Hz. These receptors have a loose plug of epithelial cells which are captively coupled to sensory receptor cells of the external environment. In the diagram, we show a fish around its electroreception in conductor or non-conductor. This is all about my diagram. This is all about my presentation. Thank you so much.